Hello students, welcome back to the class. So, today what we will be doing is, we will be talking about photosynthesis, the chapter photosynthesis in details. If you, I think you all remember in the last two classes what we have done is, we have just tried to understand the chapter. We have discussed about the summary of the chapter. Now, at this time, we have an idea about the chapter, right? Whenever we talk about photosynthesis, at least we have an idea. Photosynthesis, where does it occur, right? What is it? We are aware of the terms called as light reaction, dark reaction, words like thylakoid, stroma, etc., etc. So, these are the things that we have already discussed. Today, we will go in details and we will be starting the class, and by the introduction part of photosynthesis in the form of a note. So, everything in details. So, I would request you all to write everything what I write in the board. We will keep on writing the notes, and plus together with that we will try to explain also, right. So, let us start the chapter. So, the name of the chapter is photosynthesis in higher plants. So, let us start writing some of the points. So, we are writing some of the points. So, whenever we talk about photosynthesis, it is the most significant biochemical reaction, right, or process on which the existence of life depends. So, the next point that we will be writing is, it is an enzyme mediated by regulated anabolic and endergonic reaction. And since we are writing the notes, so and plus we have already explained, so we understand the meaning of anabolic, we understand the meaning of endergonic, right. So, photosynthesis is a reaction which is an anabolic reaction. We understand the meaning of anabolic and catabolic. Any process in which we see the formation of certain things is called as an anabolic reaction. And endergonic term was in relation with ATP, right. We had discussed already, we have two terms, endergonic reaction and exergonic reaction, right. A process in which ATP gets consumed and a process in which ATP is synthesized, right. Photosynthesis is an example of a endergonic process where the ATP gets consumed and the reaction continues, right. The next point is, in this reaction, just a second. Sorry. So, it is a process in which the light energy gets converted to chemical energy. The next line, it is an oxidation reduction reaction and whenever both the things are occurring together, we have a term that is called as redox reaction. And when we are talking about the reaction, just a second. So, since we have already mentioned it is a reaction, let us write the reaction. So, these are the few points. We have some more points to study since we are talking about the introduction part of the lesson, right. So, I think these are the sentences that we have already understood. It is an oxidation reduction reaction. Whenever we talk about photosynthesis, it is depicted by this equation, where we have carbon dioxide plus water in presence of pigments, and the pigments obviously the chlorophyll, the photosynthetic pigments together with sunlight, and there are certain more requirements also. They will be giving C6H12O6 with the evolution of oxygen and plus water. Next is, since we are talking about this as a redox reaction, I think you remember we have also explained as to who undergoes oxidation and who undergoes reduction during photosynthesis. So, this is what we are going to depict in the reaction itself. So, when we are talking about reduction, who undergoes reduction? It is carbon dioxide that undergoes reduction. And if you are talking about oxidation, who undergoes oxidation is oxidation, right. Reduction, we are talking about dark reaction. And when we are talking about oxidation, it occurs during the light reaction. So, the common equation that depicts photosynthesis, who undergoes reduction? Its carbon dioxide gets reduced to form glucose, and this is done during the dark reaction part of photosynthesis. And if you are talking about oxidation, who undergoes oxidation? It is a water molecule. We have already used a word called as photolysis of water. Obviously, we will do it in details. Photolysis of water occurs during the daytime. It is a part of the light reaction, and it results in the evolution of oxygen. The next point. %90 of total photosynthesis is carried out by aquatic plants, right. So, plants undergo photosynthesis, we understand, but maximum photosynthesis is seen by aquatic plants, and specifically we have to mention about algae. So, highest photosynthesis is seen in which group of plants? Highest photosynthesis is seen in algae, right. And again talking about algae, it is a aquatic algae. Next, the next line. The process of photosynthesis was first seen in. The process of photosynthesis was first seen in eubacteria. Eubacteria. Eubacteria is also called as key. It is also called as true bacteria. Next point. First true oxygenic photosynthesis was seen in cyanobacteria, and we know cyanobacteria is also called as what? Blue-green algae. The next point, we will be discussing this. The next point talks about photosynthesis is absent in examples. Like Cascuta, fungi, etc. Next, we also have to talk about something called as photosynthetic roots. Let me write it down first. Examples: we have some more points since we are talking about the introduction. So, these are some of the points that we have to study before starting the main chapter. Okay. So, we know that it is a reaction that undergoes oxidation and reduction. Talking about where do you see photosynthesis? Obviously, in green plants, and talking about plants point of view, we understand that there are 5 different categories of plants starting from algae to angiosperms. So, highest photosynthesis is seen in algae, right. The next is the process of photosynthesis was first seen in. We understand that photosynthesis occurs in plants, but does that mean it occurs or it is something that is seen only in case of plants? Obviously, it is seen in plants, but we have certain photosynthetic bacteria also, right. So, the process of photosynthesis was seen in case of eubacteria. And what is eubacteria? A true bacteria is called as a eubacteria. As far as eubacteria and the different types of bacteria is concerned, we have a chapter called as biological classification, and there we have a kingdom called as Monera, and we will be studying about them in details. As of now, just remember from photosynthesis point of view, you have to remember this point, that the photosynthesis was first discovered or first seen in, it was seen in a bacteria, right. And which bacteria? Obviously, a photosynthetic bacteria. Next, the next line that we have written is first true oxygenic photosynthesis was seen in. Just a second. first true oxygenic photosynthesis was seen in. These two lines talks about the thing that photosynthesis first time it was discovered or it was noticed in eubacteria, but this type of a photosynthesis was noticed in cyanobacteria. What is the difference between the first line and the second line? These two lines says that no doubt photosynthesis is seen in green plants and it is seen in certain types of bacteria or photosynthetic bacteria also, but the process of photosynthesis in bacteria and in plants is different, and the most important point of difference is one being oxygenic and the other being non-oxygenic. I mean to say that whenever photosynthesis occurs in plants, it leads to the evolution of oxygen, and so this type of a photosynthesis is called as a oxygenic photosynthesis. And in case of bacteria or in case of photosynthetic bacteria, the process of photosynthesis is seen, but it does not lead to the evolution of oxygen. What is the reason behind? We will write in details. Obviously, because of the fact that green plants would be using water, and instead of water bacteria would be using H2S for the process of photosynthesis. Don't worry about that, we will be writing it as we go on, right. As of now, is this word understood? First true oxygenic photosynthesis, it talks about the process where oxygen is involved, right. Oxygen synthesis occurs in photosynthesis that occurs in plants and not in bacterial photosynthesis. So, non-oxygenic photosynthesis is seen in bacteria, oxygenic photosynthesis is seen in green plants. I think it is clear. Next is photosynthesis occurs in plants, but we have certain examples where photosynthesis is not seen. Obviously, anything that is not green in color, anything that does not have chlorophyll or photosynthetic pigments, right, they will not be showing photosynthesis. One example being fungi, the most common example. The second example is Cuscuta. One point about Cuscuta that you have to remember is Cuscuta is an example of a parasite, and I think we understand what a parasite is, right. As far as nutrition is concerned, either an organism would be a autotroph or it will be a heterotroph. If it is an autotroph, it will be preparing its own food, and if it is a heterotroph, it will be taking the food from some other organisms, right. As far as heterotrophs is concerned, if the organism take the food from a living host, it would be considered as a parasite. If it is taking the food from a dead host, it would be considered as a saprophytes, right. So, what is Cuscuta? Cuscuta is an example of a parasite. Later we will study about the classification of parasites being, we have certain words like stem parasites, root parasites, total stem parasite, total root parasite. Together with that, we have a word called as partial root parasite, partial stem parasite, etc., etc. As of now, Cuscuta is an example of a total stem parasite, right. The next point talks about photosynthetic roots. As far as photosynthesis is concerned, where does photosynthesis occur? We understand, right, the basic answer is obviously in leaves, and what is the reason is because of the presence of chlorophyll. So, whenever we are talking about that part of the plant, we never think that roots will undergo photosynthesis. First, they are under the ground, right. So, in general, roots are under the ground and they do not have chlorophyll, but we have exceptions where we have certain examples like Ctenospora and Trappa. They would be having photosynthetic roots, and this photosynthetic roots would be, they would be called by another name. They are also called as assimilatory roots, right. So, give an example of a plant who has a photosynthetic root. So, answer would be Ctenospora and Trappa, and I think these two examples is more than sufficient to remember, right. Okay, let us go to the next point. Okay. The next point talks about have you heard this word euglena? So, 2 points about euglena. Obviously, it is a photosynthetic organism. And it shows a link between plants and animals. So, we have to mention about euglena when we are talking about photosynthesis. It is a photosynthetic organism and it also shows a link between plants and animals. By this statement, what we understand is this is an organism that would be showing certain plant-like characteristics and certain animal-like characteristics. Talking about euglena, where you are going to study about euglena in details? Again, in the chapter called as biological classification, where we are going to study the different kingdoms, right. So, euglena, I think you have already heard about a kingdom called as Protista. Right. So, we will be studying about that in details. It is, as of now, it is a photosynthetic organism that will have certain plant-like characters and it will have animal-like characteristics also. What plant-like characteristics and what are the animal-like characteristics that it show, we will be discussing in the biological classification chapter. Right. So, these are certain points that comes in the introduction of the chapter. So, we have talked about it being a reaction, who undergoes oxidation, who undergoes reduction. We have come across a word called as photosynthetic route, which is also called as assimilatory route, right. We can just write down a point. Are also called as assimilatory routes, right, just to remember. So, give an example of a plant with assimilatory route or give an example of a plant with photosynthetic routes. We remember the examples of Tinospora and Trappa. After this, what we need to study is we have to talk about the experiments and the contribution of certain scientists related with the chapter. From this part, I mean in this part, what you need to study is the names of each and every scientist that is given in your NCERT book. Together with that, each and every experiment that is given in your NCERT book. What we are going to do in the classes, we are going to write only a few of them, right. Let us start writing. So, the next heading would be certain experiments and certain name of the scientists that you need to remember or study. From NCERT also, or from NCERT you have to study, and apart from that we will be talking about just one or two important. We will be talking about the first experiment that was done using something called as a variegated leaf. Let me write down the experiment first and then we will explain. So, in the first experiment, what was done is 2 leaves were taken of a plant . So, the experiment was such that we have right. So, we had sunlight, just a rough diagram so that you understand the experiment. Right. So, the experiment was done where 2 leaves were taken, where one leaf was completely exposed by light and the other partially covered. It was partially covered by black paper, but here in the diagram, instead of drawing black, obviously it is not possible. So, what we are doing is we are showing it with a red. This is a variegated leaf. What is the meaning of a variegated leaf? We will be explaining. A leaf that has more than one color is called as a variegated leaf. So, the experiment was done as such, one leaf or one variegated leaf and the other leaf was exposed to light. The leaf that was exposed to light, later it was seen that one variegated leaf, that is let me write it down please. Okay. So, we are talking about the first experiment, the experiment with variegated leaf. First, we need to understand the meaning of variegated. A leaf that has more colors is called as a variegated leaf. In this case, obviously, we are talking about a green leaf, but since we have covered one portion with a paper, so it makes the leaf two colored. The portion that has been covered, obviously, it will be white, right? And this is, and the remaining portion would be green. So, since this is covered, it has two colored in the leaf, this leaf is called as a variegated leaf. So, what he has done is, so the scientist has taken the two leaves and both the leaves were exposed with sunlight, and whenever after keeping this experiment for some time, what they have seen, the leaf that was completely exposed to sunlight, obviously photosynthesis would occur, and as a result of photosynthesis, what we will be observing is starch formation is seen in this leaf. But in this leaf, the portion that was covered with black paper, or the portion that was covered, since it did not receive light, starch synthesis at that part was not seen. So, this is the experiment, and what is the conclusion of this experiment? Photosynthesis occurs only in the green parts of plants in presence of light. Right? So, the first experiment, the experiment with variegated leaf, the question that is asked is, what does it prove or what does it say? The experiment with variegated leaf proved that, or it concluded that photosynthesis is something that would be occurring only in the green parts of the plant. So, this is what they have concluded, and whenever we are talking about the green parts of the plant, in other way around, what we can say is photosynthesis occurs only when chlorophyll is present, and green parts of a leaf, why is that part green? Obviously, because of the presence of chlorophyll. So, this experiment concludes that if photosynthesis has to happen, then chlorophyll needs to be present. So, with respect to this experiment, one question has been asked. Let us discuss the question, so that we can have an idea as to how we need to study. When we are studying in this class, I mean when we are studying the chapter, what we will do is, we will be talking about certain questions related to that topic also, right. So, this is the experiment, and based on this a question has been asked. Let us see. Okay. What is the question, and if you have understood the experiment. I am sure you will be able to answer this also. Variegated leaf experiment So, the question goes like this. So, we have already discussed that experiment. What we have seen is starch formation is seen only in the green parts of the plant when the plant is exposed to light. We remember that leaf or that portion of the leaf which was covered with black paper, and in that portion we have not seen starch formation. So, what does this indicate? That is the portion where photosynthesis was not done, or starch formation was not seen. So, the question in relation with that experiment. So, the question goes like this: variegated leaf experiment is carried out to test the presence of. So, I think you will be able to answer. Obviously, it was not carried out, or it is an experiment that did not say anything about fructose, water, neither it talked about carbohydrates or food. So, what did it say? The main point was, if you remember, we have underlined this word, right. So, what would be the answer? The answer would be, so it is an experiment that talks about chlorophyll, right. So, this was the first experiment. Let us go to the next one. Experiment number 2. So, it was called as Moll's half leaf experiment. From MCQ point of view, again, what is the question that would be asked is, what is the conclusion of the experiment, or what did it prove? Obviously, the questions like discuss Moll's half leaf experiment, this type of a descriptive question is never going to be asked in your MCQs. So, when we are talking about Board's point of view, we are going to write about the experiment, we are going to draw the diagram, right. Each and every detail we need to write in our copies, but from studying from MCQ point of view or entrance point of view, it is mainly the conclusion and the materials that has been used that a question is generally asked about, right. So, let us write about this. So, Moll's half leaf experiment, this experiment showed that so, this experiment talks about the role of CO2. If you remember, the first experiment talked about what? It talked about the role of chlorophyll in photosynthesis. This is the experiment that would be talking about the presence of, or the requirement of carbon dioxide for photosynthesis. So, this is one important word. So, how does the experiment go? This experiment, what they have done is, they have, again, I am not writing each and every thing in sentence, just from the point of understanding. So, what they have done is, they have taken a plant and they have taken a part of a leaf Let me write it down first. And in this okay. So, we are talking about Moll's half leaf experiment. What they have done? Again, they have taken a plant, right, that was exposed to sunlight, right. So, this was leaf number 1 that was exposed to sunlight. Since it is exposed to sunlight, obviously, we will see starch formation. Just a second. This slide completely exposed to sunlight, it has all the requirements. So, we will see the formation of starch or photosynthesis occurs, right. In what they have done is, they have taken one leaf exposed to sunlight where they have seen the starch formation. So, we understood. The next is, they have taken a part of a leaf. So, what they have done is, they have cut apart this portion, they have taken a test tube. In the test tube, what they have done is, they have put just a second, let me draw it down. They have taken a test tube. In the test tube, they have taken cotton, right, and this portion that was cut, this portion, the leaf, right. So, what we have now, we have this as the cotton, right, and this cotton, what was so special? It was KOH, or it had KOH solution. Soaked cotton, and this one is this part, and they have exposed this test tube under sunlight also. Could you understand this? What they have done is, they have taken this plant, they are doing the experiment with the help of these two leaves. This leaf completely exposed to sunlight, and because of that, we know photosynthesis occur and we will see starch formation. On the other hand, what they have done is, they have cut a portion of this leaf and they have put it in a test tube, and this test tube was also exposed to light. So, if you see, the leaf is there, so obviously the green part is there, right. It is exposed to sunlight, so it is absorbing sunlight also. So, and plus we know for photosynthesis to occur, the leaf needs to take in carbon dioxide also, and from where will they take in the carbon dioxide? They would be taking in the carbon dioxide from the atmosphere. Since this leaf is conducting photosynthesis, we know that it is taking in sunlight, it is taking in atmospheric carbon dioxide, it has chlorophyll. So, all the reasons are sufficient to conduct photosynthesis. On the other hand, this leaf also has chlorophyll because it's just a cut part of the leaf. It is also exposed to sunlight, and it can also, or since it is exposed to sunlight, it has the capacity to absorb carbon dioxide also, because obviously it will be taking the carbon dioxide from the atmosphere, and we have not done anything with that carbon dioxide, or we have not prevented, or we have nothing. We have done nothing to hinder the taking in of carbon dioxide. But interestingly, what they have seen is this part, no doubt it was exposed to sunlight, it had chlorophyll, but later it was seen that the starch formation was not seen. And what could be the reason behind? They have later understood, or they later concluded that why this leaf or this part of the leaf did not show starch formation, because this part of the leaf did not absorb the atmospheric carbon dioxide. This leaf showed photosynthesis because it took in light, it took in carbon dioxide. This leaf failed to show, or this part of the leaf failed to show photosynthesis because it could not absorb the atmospheric carbon dioxide. And why it could not absorb the atmospheric carbon dioxide? Because later it was seen that when we are talking about this cotton had what? KOH soaked cotton. So, we know that KOH is a good acceptor of carbon dioxide. So, what it has done, all the atmospheric carbon dioxide was absorbed by this cotton, and as a result the leaf was unable to absorb it. So, it could only take in light, it had chlorophyll, it could take in light, but it could not absorb carbon dioxide, and that is the reason this is a part of the leaf, or this part of the leaf did not show photosynthesis. And this experiment talks about that fact, that until and unless carbon dioxide is absorbed, photosynthesis will not occur. So, this is what is called as Moll's half-leaf experiment, which concluded that carbon dioxide is essential for photosynthesis. So, we have discussed today in the class two experiments. The first was with the variegated leaf, which showed the importance of chlorophyll in photosynthesis, and this is Moll's half-leaf, which showed the importance of carbon dioxide in photosynthesis. So, again let us do a question that has been asked from this part. One common question that has been asked, let us not write that down. The common question that is asked is, what does Moll's half-leaf experiment conclude? It concludes that carbon dioxide is essential for photosynthesis. So, that is something that is very common. So, we are not going to write it down. Another question that has been asked from this point, let us write down that question. Let us see whether we can answer or not. So, the question goes like this: carbon dioxide is necessary for photosynthesis the chemical used to remove this gas more efficiently from entering a control apparatus is. So, please read the question properly. Calcium oxide, distilled water, potassium hydroxide and option D is sodium carbonate. Okay, so this is the question that was asked from that part. As I have said, I have not written the most common question that is asked. What was the most common question? What does Moll's half-leaf experiment talks about? It talks about the importance of carbon dioxide in photosynthesis. Another question from that part, the question goes like this: carbon dioxide is necessary for photosynthesis. This is something that everyone knows about it. The chemical, so the question is about the chemical that is used to remove this gas from entering into the controlled apparatus. By the word controlled apparatus, we are talking about what? If you remember, we have talked about this test tube, right? So, this is something that we are doing. So, it is being mentioned as the controlled apparatus. So, what is the solution that was preventing the absorption of carbon dioxide? Read the question and see whether you have understood or not. Carbon dioxide is necessary for photosynthesis. The chemical that is used to remove the gas. So, we are talking about this one, the chemical that was used to remove the gas from gas more efficiently from entering a controlled apparatus, right. So, who was responsible? Obviously, potassium hydroxide solution. We have talked about what? If you remember, we have written this now. Cotton in the experiment, right. We have talked about this experiment. We have used KOH soaked cotton, and it is because of KOH that the leaf was not being able to absorb carbon dioxide. I think it's clear. So, these are the two experiments. So, together with that, we also have to talk about bell jar experiment that was conducted by Priestley, right. So, that is an experiment with the diagrams itself. It has been given clearly in your NCERT book. So, we will be talking about, or we will be referring to the diagram from the NCERT book. So, experiment wise, we will be discussing two of them, and let us go to the scientist part now. Contribution of some scientists. This is something that we are going to write it down. Sorry. So, this is in the form of a note. So, first we will be talking about Stephen Hales. Next, Joseph Priestley. what is the experiment that he has done. He has done the Belger experiment, and in this experiment he has used a Belger, right, what did he conclude? He talked about purification of air. So, talking about this part where we are writing the contribution of certain scientists, certain questions, I mean direct questions from the information that we have written, especially question has been asked about this one Belger experiment. Who has given the Belger experiment? You have to know the name of the scientist. Again the question has been asked about, once the question was asked which plant was used in this experiment. So, the name of the plant is also important, and obviously the conclusion, or the question could be asked talking about exchange of gases or gaseous exchange was something that was dealt in Belger experiment. It was conducted by Presley, or what did Presley talk about? Presley talked about exchange of gases, or Presley has talked about the purification of air. So, this is how we are going to study, right. The next scientist Van Niel. He has given the concept of, or he had discovered bacterial photosynthesis. Next, Rubin and Kamen. So, what is the contribution of Rubin and Kamen? So, again from this point, who has discovered bacterial photosynthesis? Van Niel has discovered bacterial photosynthesis. Details about bacterial photosynthesis as we go along the chapter we will be discussing later. Next, the contribution of the two scientists, what they have said. They said that so, this is one important line. So, we see the evolution of oxygen, carbon dioxide plus water will give you the food and the oxygen. So, oxygen is released from, or who is responsible? Obviously, the water molecule, because previously the scientists were people used to think that the carbon dioxide used to come out from the oxygen used to come out from carbon dioxide and not from water, but this experiment proved that it is water who is responsible for the evolution of oxygen and not carbon dioxide during the process of photosynthesis. Next, while they went to prove this, they had used a radioactive isotope of oxygen. This point is also important. That is oxygen 18. Very important. So, what they have done? Okay. So, Rubin and Kamin, this is something that's very, sorry, it's important again. So, Rubin and Kamin, what is important is what they have said is they have concluded that oxygen gets evolved from water by using what they have used a radioactive isotope of oxygen. Oxygen 18 is something that you need to remember. This is what they have done. So, oxygen 16 here, and with this 18, if you are talking about this one, if you look into this, we will see that this is the one that is present in this one. So, with this one, I mean from this point a question has been asked. Let us do the question, and this will be the last for today. So, what is the question that is asked from here? Okay. If green plant cells are incubated with oxygen 18 labeled water, if green plant cells are incubated with oxygen 18 labeled water, which of the following molecule will become radioactive when the cells are exposed to light? The options are okay. So, read the question. In green plant cell, if green plant cells are incubated with O18 labeled water, which of the following molecule will become radioactive when the cells are exposed to light? So, obviously, this question is directly asked from this. So, what is the see, whenever you read the question, at first you might see that it consists of many sentences together, but if you read it properly, it is something that is very simple, or the question is about the concept that we have studied. So, do not get frightened by reading all the sentences, right. No doubt there are 3-4 sentences, but the answer is easy because we have already studied about the contribution of Rubin and Kamin, right. What do you remember about Rubin and Kamin? First, oxygen comes from water. Second, he has proved it by using what? Radioactive isotope of oxygen, that is oxygen 18. So, which molecule would be becoming this? Obviously, the answer would be this one. So, just go through the question and see whether you have understood or not, right. So, today in the class, I think this is all for today. What we have done today is we have started this chapter in details. We are starting to write the notes, right, and we are talking about the contribution of the different scientists, right. We have not completed this portion. We are going to continue it in the next class. Today up till here, I think this is all, and together with the contribution, we also had an idea as to what are the questions or what are the type of questions that are being asked, and this is the way that you have to study. Anyway, thank you for today. We will be continuing with in the next class. Thank you.